Hi, my name's Phoebe Griffith and I'm a crocodilian conservationist. So crocodilian is the group that encompasses the crocodiles, the alligators and caimans, and the gharial. And it's the gharial that I specialise in. Gharial aren't as familiar to most people as crocodiles and alligators, but they're really special and they're a lot weirder. So the gharial is a really unique species. And they're not in fact that closely related to crocodiles. In fact, they're only as closely related to crocodiles as we humans are to lemurs. So they're really evolutionarily unique. They're kind of all alone on their own branch in the tree of life. And I have here my plush gharial to help me illustrate some of the things that are special about a gharial. So the first is they have this really long, thin snout, which is specialised for catching underwater prey, mostly fish, but they will also take things like turtles and crustaceans. And it's actually a very fragile snout, which means that they're fairly safe to people. They don't attack people. And they're also super adapted for life in the rivers. So the gharial has this broad tail for propelling itself through the water. And unlike crocs or alligators, which some of you may have seen on YouTube strolling across golf courses, gharials can't stand high up on their legs like other crocs can. Which means they're much more limited to the water. So they spend almost all their time in the water or directly on the sandbanks beside the river. And now this gharial may be small, but they can in fact grow huge. Although they're born at only about 30 centimetres, they can eventually grow over 6 metres long. And we suspect historically some gharial got up to 8 metres. So this is an adult female gharial. And as you will notice, she has babies on her head, and that's because gharial are great parents. Both the male and female gharial will look after the babies while they're very small, because at this time they're in fact a tasty snack to a variety of different wildlife. And this is an adult male, and you will notice he has a weird lump on the end of his snout. And this is in fact unique to gharial and unique to the adult males. And it's called a gara, which is a word for a clay pot in Hindi. And it's what the word gharial comes from. And the males probably use this for amplifying vocal communications. So the reason I'm a gharial conservationist is that gharial are critically endangered, which means they're at imminent risk of extinction in the wild. We've in fact, since the 1950s, seen 94% or more declines in gharial numbers in the wild, and they now only occur in India and Nepal. And I'm very lucky to be able to work on gharial in Nepal. And we're investigating as much as we can about them so that we can inform their conservation strategy and hopefully increase numbers again. So one problem with gharial is that we really struggle to tell individual gharial apart, which can be tricky for monitoring populations. For example, if we see two gharial basking and one disappears underwater and then an individual reappears, have we got a group of two gharial? Or is this in fact a new individual and we have a group of 3? Also, if we can't tell individual gharial apart, we can't be sure which gharial is doing an activity, say nesting, and whether that's the same individual we saw basking in a different area a month, an hour, or a day earlier. Another major question is how much of the river do gharial use? If we see gharial across a 100-kilometre stretch of river, are all the individual gharial using all of the river? Or is each gharial using just a small stretch of the river? Or perhaps their strategy differs according to the size of the gharial? So the way we're answering some of these questions is through using radio telemetry. So we put radio tracking devices on the wild gharial in order to answer some of these questions about gharial river use, about their habitat choices, and about causes of mortality, which means the different ways in which they're dying. So how do you put a radio on a croc? So we attach the radio to the gharial's tail while it's blindfolded and restrained, and then we release the gharial and she'll go straight back into the river once the blindfold is removed. And the whole process takes about half an hour. We can then track her movements in the wild for the duration of the radio battery life, which is usually about 18 months to 2 years. So once we've put these radio tracking devices on the wild gharial, we then have to go out alongside the river to find out where the gharial are. And so this requires going out with a big antenna. So this little purple person is me, and that's me in real life. So we go alongside the riverbank, tracking where the gharial are, which have their own unique radio on. So here I am with my antenna, and what we're doing is we're actually listening for this unique beep beep frequency that's released by the radio and gets picked up by the antenna. And that means we can locate where the gharial is, whether it's underwater and we can't see it, or if it's on the bank. And if it moves, we can pick up the signal in a different location. So we can know that if that specific gharial's moved, because we know the different individual frequencies of the radio. The other awesome thing that radio telemetry allows us to do is we can then identify each individual gharial. So as well as knowing exactly how much of the river that individual gharial is using, we can see what particular activities it's carrying out. So as we track these animals over 2 years, we can see where they're choosing to bask, whether they're nesting and mating, where they're nesting, and how that might relate to the ways they use the river at other times of year. And the way we're able to do this is because of each individual radio having its own frequency. So we may see a gharial basking and we can pick up and detect that that's, say, gharial 2, but we could get a different frequency coming and we can recognise that that is a different gharial that may be underwater and not visible. Or it may become visible, it may then move, and we can continue to track it and understand how they're moving relative to each other. Then they may both disappear, and at a later date we may detect the same one in a different location, but we know whether that's the same gharial moving downstream or if we're just seeing another gharial. So how all this comes together is we use this data from the gharial movements to build up really accurate maps of gharial river use. So we identify how the gharial are using different habitats and areas of the river and how that differs throughout the year and in different seasons. It also enables us to identify key areas that are important, for example, areas of nesting or areas of refuge during floods. One of the exciting things we found out is that because we can track the gharials during the monsoon when they spend most of the time underwater and are therefore not very visible, we found that during this time they're actually travelling outside of the national park and going considerable distance up small rivers into agricultural areas. So we as conservationists can now prioritise looking at ways to help gharials survive and thrive in these areas they're choosing to go into. So that's a little bit about my role as a conservation scientist, using science to inform conservation to help the gharial recover from the brink of extinction. So hopefully they won't be endangered in the future.
Hello, my name is Michelle and I'm a computational biochemist and senior lecturer at the University of Roehampton, London. And I'm here today to talk about my work on the effects of addictive drugs on the brain. So before I start telling you about my research in this field, I guess it would be important to explain what a computational biochemist does. The first part of that, though, is to address what is a biochemist. So generally speaking, biochemists study the chemical and physical principles of living things and of biological processes, such as cell development, growth, heredity, and disease. And a computational biochemist uses computers to study these processes. These computers are not necessarily like the desktop computers or laptops that you use, although you can work on them for analysis or running small jobs. Instead, I use big specialized supercomputers like these ones that are specifically designed for the computations that I need to perform. So how do I study the effects of addictive drugs on the brain with these types of computers? We first need to get introduced to the brain, and what better way with some giant microbe models. Here, deep within the brain is a set of structures called the limbic system, which contains the brain's reward pathway, and it is responsible for our basic survival instincts. So when you do essential things to stay alive, like drink, eat, find shelter, build relationships, or care for people, your brain reinforces these behaviors by causing the release of dopamine and serotonin from this region. The reward for surviving is also transmitted to other portions of the brain in that area, like the amygdala and hippocampus, which records a memory of that feeling, so we seek it again because we feel happy. Take, for example, if someone gave you this delicious and large donut. I'm sure after you finished eating it, you may feel that craving to have another. This is our survival hardwiring at work. When the reward pathway is activated, each individual cell or neurons like this in the pathway relays electrical and chemical signals. These neuronal cells do this because of their structure, having a large body and a long tail or axon at the end with little branches that can communicate with other cells. In this figure, you can see the reward pathway in the brain on the left, and on the right, you can see that the small gap between connected neurons is called a synapse. In the reward pathway, neurons release the neurotransmitter dopamine, depicted here as little blue dots. The released dopamine molecules travel across the synapse and bind to proteins called dopamine receptors, depicted here as purple blobs on the surface of the receiving cell or postsynaptic cell. This causes these receptors to carry the signal onward within the cell, as shown by the arrows. Some dopamine molecules re-enter the sending cell or presynaptic cell via dopamine transporters, shown as green structures, and they can be released. When a reward is encountered, the presynaptic cell releases a large amount of dopamine in a sudden burst. Dopamine transporters will then quickly remove the excess. It's important to note that dopamine surges in response to natural rewards help the brain learn and adapt to a complex world. However, drugs like cocaine are able to hijack this reward pathway, affecting parts of the brain in the front, like the prefrontal cortex, that controls decision making and impulse control and contributes to unhealthy behaviors and consequences. It does so by making you believe that the drug is now instead your primary need for survival. Scary, huh? When someone first uses cocaine, the drug quickly enters the brain, where it blocks the transporters on the presynaptic cell, depicted here by the red X's. Since dopamine cannot re-enter the presynaptic cell, it begins to accumulate in the synapse, where it can reach abnormally high levels and remain there much longer than usual. The postsynaptic cell becomes overactivated, which produces a feeling of increased happiness. This creates an incredibly powerful association between cocaine and pleasure, making a person want to repeat the experience of taking the drug. I am sure you can agree that making you believe that you are dependent on a drug for survival is a pretty dastardly feat for your brain to accomplish. But drugs like cocaine, amphetamine, and new designer drugs, like new psychoactive substances, can also cause the brain tissue to become increasingly damaged with continued use, and this in itself is not good. So lots of scientists are working at pace to understand the effects of these drugs that are becoming increasingly available to people. This is because even though they may structurally look like known illegal drugs like cocaine and amphetamine, their pharmacology and toxicology are unknown. This is because these drugs are made in unsafe laboratories and by people who only think of profit rather than people's health. But it is up to all of us to be educated about the use of drugs and how they affect us. So in my work, I want to understand whether these new compounds shown here, and also in their form that you would buy them on the street, whether they block the reuptake of dopamine from the dopamine transporter like cocaine or release dopamine like amphetamine. I design models of these drugs and transporters on the computer and investigate through the use of mathematics, physics, biochemistry, and even computer science, and by using supercomputers to understand how these drugs interact with transporters. In the end, I can make really lovely animated movies, like this one here, of these computations to see structurally how and where these drugs bind to the transporters, or even the different conformations that the transporter can adopt when transporting dopamine. We can use this information to identify at the molecular level the different pharmacological actions that we see in real life, and that's how I use computers to study addiction and the brain. Thank you very much for listening.
Hi, everybody. My name is Andrea Townsend-Nicholson, and I'm a professor at UCL, and I'd like to describe a really exciting project that my colleagues and I are working on building a virtual human. And we want to build a virtual human for every single person on the planet. So what is a virtual human? Well, it's an exact replica of you. identical at every level of detail, all the way from your DNA sequence up to the structure of your skeleton and how it moves. It is a perfect digital equivalent of your analog self. And why would you want to have a virtual human? Why do you need a digital twin, or why would you want to have a digital twin? Well, they can be incredibly useful. They're a very good way of being able to manage your health, your well-being, and your fitness. So for example, if you have a particular health problem, your doctor can try out many different treatments on your digital twin to choose the one that will work best for you, and then they can give you that treatment. If you want to improve your fitness, or if you want to improve your lifestyle or your general well-being, you can try different nutrition plans, different diets, different exercise programs on your digital twin and pick the one that works best on them, knowing that because they are identical to you, that will be what works best for you. And if you really, really, really want to develop yourself to the best that you possibly can be. Let's say you're an athlete and you want to have optimal performance. You can try different things on your digital twin, on your digital avatar. So you can work out exercise plans, rest plans, nutrition programs, the way in which you try different things as part of your working routine to develop your athletic ability and to develop your resistance and your stamina. And you can see what works best on your digital twin and use it for yourself to optimize your athletic performance so that you can be better, faster, fitter, stronger, depending on what you need for the area in which you wish to be developing your skill. So how do you build a virtual human? What goes into a virtual human? Well, it's a digital twin and it's built inside a computer. So it's going to have lots of data that go together. Where you are flesh and bone, it will be bits and bytes. And so different pieces of data are coming together. So what kinds of data go into a digital twin? Because it's built inside a computer, so it's going to be built of data. We can take a look at many different kinds. I'll start with a few here. Your DNA. Your DNA sequence is unique to you, and your virtual twin will be built from the DNA sequence that you have. It will also have data that includes images, scans of your organs, your heart, for example, or your lungs. It can include data like your skeleton from an X-ray. It can also include information about the way in which your skeleton moves. So you put the little dots on somebody and a camera on them, and a computer works out the way in which they move and they map that onto the skeleton. We've actually made virtual skeletons dance in the exact same way that the physical person dances. So they're an exact replica of each other. There are some bits of the virtual human that we have already built. So this example of the dancing skeleton actually has a more practical application behind it. You can take a look at how somebody's skeleton moves. You can look at their bone mineral density, so you know how strong their bones are and where there are weaknesses in their bones. And you can use this information to work out with movement where there are weaknesses in the bones and where there might be potential fractures as the person ages. And then you can help do things to make the probability that that happens less, so that people are less likely to develop a fracture. We can take information about a heart and the electrical pattern, the beating of the heart and its structure, and we can work out where to put a pacemaker for a patient who needs to have a pacemaker to make sure that their heart rhythm is restored to normal. If somebody has a blocked blood vessel in their heart, you can use this kind of information to work out where to put little devices called stents that will be able to open up the vessel so that the blood flow can go through more easily. So not only can we make a digital twin dance in a supercomputer, we can make a virtual heart beat in a supercomputer. We even have a supercomputer that can tell whether a heart is male or female when we build a digital twin heart inside it. Some of the things that we can't quite do yet, we can't do because we can't do them fast enough. So the area that my lab is particularly interested in working on is the interaction between the atoms of the proteins in your cells and the atoms of drugs that can be used to make those proteins work better. If you have particular conditions where you need some kind of a drug treatment for it, we look at the protein that needs to be treated, and we look to find the best drug for that treatment for that person. And sometimes there are small variations in proteins between people, so we can actually choose or make the drug that is best for that particular person's specific protein. This is also particularly important right now because we, along with tens of thousands of other labs across the world, are very interested in finding out what drugs will be able to interact with specific proteins of the virus known as SARS-CoV-2, which causes the disease COVID-19. And we are all trying to find the best match so that we can find drugs that prevent SARS-CoV-2 from causing COVID-19. So that's a practical application of something relevant right now that we're all trying to do. But more broadly, we're interested in finding the best match between protein atoms and drug atoms to help improve people's health. So, when can we expect to see a virtual human? When are we going to have our first digital twins appearing, and when will we be able to test all these different things on them, test new drug treatments, have our doctors work with them, have ourselves work with them, maybe our coaches and our athletic teams work with our digital twins? Well, we're some way off right now. It won't be for a number of years yet, and there are two specific reasons for this. One of the reasons has to do with the power of the computers that we use. So if you are building a virtual human, every component of that virtual human needs to be able to be put together, built, and needs to work on the same time scale as the person. Your doctor, when they're using your digital twin to test treatments on, needs to be able to get an answer to these different tests quickly enough that they can treat you with the information. And currently, the computers that we have are not yet sufficiently powerful for this. We can do these calculations, we can build these parts of the virtual human, but they don't work fast enough to be able to be used by our doctors. However, we're in the midst of building the next generation of supercomputers, exascale computers, and these will start showing up in the next 2 to 3 years, and they are 1,000 times faster than anything we currently have, and this will allow us to do these atoms interacting with atoms parts of the virtual human at a speed that's fast enough to be useful for our doctors. And because we can do those components that are part of the virtual human at the right speed, we will be able to do the whole thing at the right speed, building an entire digital twin for each of us. So I said that there was one thing, that was the computers, but there was a second thing, and that is you. We need people who are passionate about helping build a virtual human. We need people to be biologists, medical doctors, nurses, mathematicians, physicists, computer scientists, engineers. We need everybody to come join us and help us build these virtual humans better, faster, more accurate. So whether you want to watch us build them, or even better, join us in building them, please keep an eye out, because a virtual human, possibly your own, will be coming soon, and you'll be able to see exactly how they can change your life for the better. Thank you.
A brief background about me. I'm a climate scientist. I did my PhD from Imperial College London in atmospheric physics. Since then, I worked in various research organizations including Imperial College, University of Exeter, etc., and also previously worked for India Meteorological Department. Why I will discuss major modes of climate variability.
because they control the weather and climate of various regions of the globe. I will familiarize you with names of various modes of climate variability. Some of the names you have already heard before. What regions are affected by those modes? Then I will talk briefly about El Nino and La Nina. Many of you have already heard those names. Then I will talk what is Indian Ocean Dipole, and then some effects in various countries due to those modes. Here are names of major modes of climate variability around surface level. Here is the plot of the whole of the globe. I'm going to show how different regions are affected by various modes that I mentioned earlier. Let us first talk about ENSO. Can you tell what region is ENSO mainly influencing? It is mainly present in the region of Central to Eastern Pacific Ocean. Indian Ocean Dipole is present in the Indian Ocean region, whereas Indian summer monsoon is influencing the whole Indian subcontinent. Now let us focus on ENSO. There are two plots. In both the plots, we are showing certain portion of the globe that covers Pacific Ocean. In each plot, the right-hand side shows Northern America and Southern America, while the left-hand side it is showing the Australia and other countries. In the ocean water, when it is warmer than normal, it is shown by red color, while it is cooler than normal, it is shown by blue color. In the left-hand plot, you can see there is a strong warming from central to eastern equatorial Pacific region. And if that situation happens, then it is known as warm event of ENSO or El Nino. In the right hand side, you can see that there is a cooling as shown by blue color from central to eastern equatorial Pacific, and it is known as cold event type of ENSO or La Nina. Hence, we can discuss what do you mean by El Nino. At the bottom I have written here. The El Nino or warm event of ENSO is the periodic warming that occurs in the central to eastern tropical Pacific. It adversely affects the fishing industry around the coast of Peru and Ecuador, while during La Nina or cold event, there is cooling. Now anyone who knows Spanish, they can tell the meaning of El Nino. El Nino in Spanish means Christ child because ENSO usually peaks around the time of Christmas. And in Spanish, boy child is known as El Nino and girl child is called La Nina. Here is a time series plot starting from 1950 of ONI, which is Central Pacific Ocean temperature. When the ocean temperature is positive, it is shown by red color, and those years are called warm event of ENSO or El Nino. While the ocean temperatures are cold, it is termed as La Nina or cold event of ENSO and shown here by blue. If those temperature exceeds certain threshold, they're termed moderate, strong, and very strong cases. Now, can anyone tell which years were very strong El Nino? Now, here you can see those were 1982-83, 1997-98, and 2015-16. Though ENSO is originated in the Central Pacific Ocean, but the effect of ENSO is felt almost all part of the globe. Here I am showing the influence of ENSO during June, July, August. Warm region during El Nino are shown by pink color, while cold regions are by sky blue. The green color shows wet region and yellow the dry regions. Now from this plot you can see what regions are affected in what way. Now can you tell what usually happens in India during June, July, August? First you have to spot the region of India, and you can see in the Indian region it is marked by pink and yellow color. Then what will happen? It should be warm and dry. If it is warm and dry, that means most of the drought years in India are usually associated with El Nino. What about Australia? It is marked by yellow. Eastern part of Australia is very high during El Nino. And what is the consequences? One consequence is very prone to forest fire during El Nino years, and hence extra precaution is necessary. Here are plots that shows the influence of ENSO is not exactly opposite for El Nino to that from La Nina, and also the effects are not similar or opposite in winter to that from summer. The top plot is for December, January, February shown by DJF. The bottom plot is for June, July, August shown by JJA, while right hand side is for La Nina and left hand side for El Nino. From those plots it is clear that not same effect is seen in winter and summer, and not opposite for El Nino from La Nina. If you have time, you just go through the places and can find what can happen during El Nino or La Nina, or if it is a summer or winter, what will be the effect. This is the current state of ENSO. It is from May to August. What is happening in the central to eastern Pacific? You can see there is a cooling shown by blue color. And if it is cooling, that means it is cold event type of ENSO. And then what will happen in all over the globe? You can just figure it out from the plot that I showed earlier. Earlier we discussed ENSO is having a strong influence on India as well as in Australia. Here I'm showing that Indian Ocean dipole, another major source of variability that is present in Indian Ocean region, is having a very strong influence for controlling weather and climate of those two countries. For positive phase, India, Australia and Africa, all three countries are affected in one way, while the effect is reverse for negative phase. For positive phase, Darwin of Australia has very cold ocean water and it experiences drought kind of situation, while in India there is a heavy rain. For negative phase, on the contrary, there is a warm ocean water in Darwin of Australia associated with heavy rain, while there is a drought in India. Now, Indian Ocean Dipole Index is measured by sea surface temperature difference between the western equatorial Indian Ocean region to that from southeastern equatorial Indian Ocean region. That means both ends of the Indian Ocean temperature difference gives the Indian Ocean dipole value. Here is the time series plot of Indian Ocean dipole. Sometimes it is positive, sometimes it is negative. And during 2019, you can see from July onward, it started rising very high. It was an unusual high value of positive side of IOD. Now, when it is very positive value, Australia becomes very dry. That means it is very prone to bushfire. And during 2019, there was an unusual forest fire happened in Australia, and simultaneously the ENSO phase, it was again positive, and positive ENSO also helps dry condition in Australia, as I discussed before. That means 2019 July onward, the forest fire that experienced in Australia, Indian Ocean Dipole and ENSO both had some roles.